Hello students, I am Mohan Kumari. Welcome back to MK classes for biology. Today I am going to solve few of the NEET questions of the chapter Reproduction in Organisms. Let us see the first question. Androgenic haploids are useful in plant breeding. So our options are to produce better hybrids, to produce homozygous organisms, to produce pathogen-free organism, or none of these. So here androgenic haploids comes in the anther culture. Yes, anther culture is done for the production of these haploids because anther contain haploid microspores which can be used to generate new plants. Such haploids produced are called androgenic haploids. These haploid plants are useful in breeding plants, so they can be used to generate genetic homozygotes. This can be done by one generation doubling of chromosomes using colchicine. So here the spindle formation can be prevented by using the colchicine same. So the correct option is to produce homozygous organism, that is the B option. So let us see the second question. So second question here, the haploid plant can be obtained by culturing. So our options are young leaves, endosperm, pollen grains, root tips. So young leaf consists of, each cell consists of diploid set of chromosomes. Endosperm consists of triploid. Pollen grains only, which consist of haploid set of chromosomes. This can be used to produce haploid plants. So root tips consist of again diploid set of chromosomes. So our correct answer is pollen grains. Let us see the third question. In which one pair both the parents can be vegetatively propagated by leaf pieces? So the options are bryophyllum and kalanchoe, chrysanthemum and agave, agave and kalanchoe, asparagus and bryophyllum. So in bryophyllum and kalanchoe, the plants can vegetatively propagate while the leaves still are attached to the branch. The leaves of this plant begin the process of mitosis and asexually reproduce in the form of plantlets that cover the edges of each leaf. Once these plantlets develop, they drop off and grow into new plants when they find fertile ground. So the correct answer is A option, that is bryophyllum and kalanchoe. Let us see the fourth question. They are means of vegetative propagation. So here we have four options. So first one is protoplast fusion, second is tissue culture, third is in vitro fertilization, D is both A and B. So here vegetative reproduction or propagation means reproduction without the involvement of sex cells or gametes. The process of vegetative reproduction can be, we had studied, that can be natural or artificial. Here protoplast fusion and plant tissue culture are the two biotechnological techniques used in the propagation of plants through vegetative cells. In case of protoplast fusion, the protoplasm of two vegetative cells are fused, thus sex cells are not involved in this. So in case of tissue culture, whole plants can be grown using a small explant made up of the meristematic tissue. So the correct option is both protoplast fusion and tissue culture. So answer is D option. Let us see the fifth question. Virus-free culture of banana can be raised from. A option is apical cells, B is pith of stem, C is leaf lamina, and D is the primary root. So in meristem culture, meristem of the plant can be cultured. So here virus-free culture can be called as it can be seen in the meristem culture. So the meristem of the plant can be used during this culturing. Generally, Shoot apical meristem and nodes are used as explant in this culture. These are grown in CK rich medium, that is cytokinin rich medium. This hormone suppresses apical dominance, resulting in the growth of the lateral branches. If several branches arise from the explant, individual shoots are separated and cultured. So here the meristem culture produces virus-free clones. So this meristematic tissue can be seen in the apical cells. So the answer is the apical cells, option A. Let us see the next question. The fertilization in rabbit and humans takes place in. So the options are A. Uterus, B. Fallopian tube, C. Cervix, and D. Ovaries. So the fertilization takes place at the junction of isthmus and ampulla. The edges of the infundibulum possesses finger-like projections called fimbriae, which helps in the collection of the ovum after ovulation. The infundibulum leads to a wider part of the oviduct or the fallopian tube called the ampulla. The last part of the oviduct, the isthmus, has a narrow lumen and it joins the uterus. During copulation, semen is released by the penis into the vagina. The motile sperm swim rapidly, pass through cervix, enter into the uterus, and finally reach the junction of isthmus and ampulla. That is called as ampullary isthmic junction of the fallopian tube. The ovum released by the ovary is also transported to the same junction, that is ampullary isthmic junction, where the actual fertilization process takes place in the fallopian tube. So the correct answer is the B option, that is the fallopian tube. Let us see the next question. Development of root from unfertilized ovary is called. As we all studied this, development of root from unfertilized ovary. So ovary developing into fruit after fertilization. Here unfertilized ovary develops into fruit. That phenomena is called as the parthenocarpy. So option is D. Right answer is parthenocarpy. So what is porogamy? So let us see all these phenomena. Porogamy, it is the fertilization of a seed plant involving the passage of the pollen tube into the ovule by the micropyle. So apospory is the development of diploid gametophytes without meiosis and spores from vegetative or non-reproductive cells of the sporophyte. Apogamy means development of haploid sporophyte without gametes and syngamy from vegetative cells of the gametophyte. So here the development of fruit from unfertilized ovary is called as the parthenocarpy, that is the D option. Let us see the next question. Development of prothallus from sporangial stalk of a fern is an example of. So if I say in normal life cycle, the diploid sporophyte of fern, the diploid sporophyte generation produces haploid spores by meiosis. The haploid spores germinate to produce haploid gametophyte, which produces haploid gametophyte by mitosis. The gamete fuses through the process of syngamy to form the diploid zygote, and the diploid zygote give rise to the diploid sporophyte. This is how life cycle takes place in the fern. In apospory, the gametophyte, so-called prothallus gametophyte, develops directly from sporophyte, that is sporangial stalk is given here, without the involvement of haploid spores. So here the answer is apospory. A option is the correct answer, apospory. Let us see the next question. In the prothallus of vascular cryptogam, The anthrozoids and eggs mature at different times as a result. So options are: there is high degree of sterility. B option, one can conclude that the plant is apomictic. C option, self-fertilization is prevented. D option, there is no change in success rate of fertilization. So, in homosporous vascular cryptogams like ferns, single type of spores are produced which give rise to haploid gametophyte on germination. The haploid gametophyte produce both male and female gametes, but cross fertilization occurs here because of the fact that the archegonia and antheridia mature at different times. So here, because of this process, self-fertilization is prevented. It has to undergo cross-fertilization. So for this, answer is C option, self-fertilization is prevented. So let us see the next question. An ovary in a non-pregnant woman will normally produce dash number of eggs in a year. So dash number of eggs which are produced from a normal non-pregnant woman normally. So human females have a pair of ovaries. Both these ovaries are functional and are capable of producing eggs here in this case. Normally, each ovary can produce a single egg or ovum each month. This way, a single ovary can produce a total of 6 eggs in a year. So, answer is B option, that is 6. So, let us see the next question. When the plant sporophyte develops to undergo maturation, it produces. So, options given: gyme, protonema, sporophyll, egg. So, here the sporophyll means leaf bearing spores. So, here the word maturation refers to the process of sexual reproduction in plants. During the process of sexual reproduction in higher plants like pteridophytes and gymnosperms, cones are produced which are the aggregates of these sporophylls. So these sporophyll may be microsporophylls or megasporophylls. So the plant sporophyte develops to undergo maturation. It has to produce spore. It produces the sporophyll. So this sporophyll bears the micro or mega spores. So correct answer is C option, that is sporophyll. Let us see the next question. During plant grafting, connection is made between stalk and scion through. So we had studied plant grafting, artificial method of vegetative propagation. So during this plant grafting, so scion and stalks are used and are connected. So grafting is a type of artificial vegetative propagation in plants. A shoot of one plant is called scion is attached to the another plant, that is stalk. So here the scion and shoot which join to each other with the help of vascular cambium. So here the vascular cambium is the answer. So the answer is cambium. During the plant grafting, connection is made between stalk and scion through the cambium, where you can see the meristematic cells and its activity. So the next one is, the plant tissue can be separated into single cells by using. So options are cellulase and pectinase, cellulase and auxins, cellulase and peg, auxins and cytokinins. The plant tissues which are surrounded with cellulosic cell wall. Between adjacent plant cell is present middle lamella. So the middle lamella is made up of calcium and pectin. Hence the plant cells of tissue are separated from each other with the help of cellulase and pectinase enzyme. So the answer is A option, cellulase and pectinase. Let us see the next question. Any cell, tissue or organ removed from a plant for the purpose of culturing is called as the explant. So this can be used in tissue culture. Explant is the correct answer. C option. Let us see the next question. Artificial induction of roots on stem before it is separated from the parent plant for propagation is called as the layering. So answer is D option, that is layering. We have studied the artificial method of vegetative propagation in that. So layering, which is artificial where the roots are artificially induced on stem before it is separated from the parent plant. So this technique is called as the layering. So this is about the today's class. So in this class we had solved about 15 questions of NEET. So in next class let me solve the further more important questions. Thank you.